So we're going to talk about chapter 2, which deals with what?
Cell.
The cell, right? And you can see here that the cell is the structural unit of living organisms. Okay? What it simply means is that you and I are made of millions and if not even billions of cells. Okay? So we said chemicals, when they group together, they form cells. And when cells group together, they form what?
Organs.
Tissues first. And tissues, when they group together, they form organs. And with organs grouped together, you have organ systems. And then organ systems together will form the entire organism. Okay? Are we good? Okay. You recording?
Yeah.
REC? Okay, good. To be sure, I might be talking for nothing. Okay. So, there are trillions of cells. All these are the building blocks of plants and animals. We know that if the cells divide, we call that mitosis. In order, let's say you have a wound in the skin here, the cells divide to replace the dead cells, right? And that's important. There are the structural units of the human body. Now, there are two types of sex cells. Obviously, the sperm is one of them. There's a male gamete. Gamete means sex cell, G-A-M-E-T-E. At the same time, the female gamete is your egg, or what we call the ovum, or what we call the oocyte in females. So these are called sex cells. All the rest of the cells are called somatic cells. It comes from the word soma, which means body. Like example, the cells in your skin, the cells in your muscles, the cells in your bone, okay? The other organs. But when it comes to sex cells, they only have two, sperm and the ovum or the oocyte. So these are all cells that are not sex cells. So this is an example of an oocyte surrounded by what?
Sperm.
Sperm. See, look at those sperms there. It takes only one to penetrate the egg in order to fertilize the egg. Right? So remember, during the sexual act, this is the ovary, this is the uterus, the fundus, the fallopian tube, or what we call the oviduct. This is the vagina or the birth canal. The sperm travels here, reaches the egg here, and then once the egg is fertilized, the sperm may travel here. The fertilized egg now becomes the zygote that will implant into the uterine wall. Okay? So you have the developing fetus there, developing. Okay? So here is an example of the word simple squamous. And simple squamous means simple means one layer. And like when you hear the word stratified, stratified means what? Two or more layers of cells. The word squamous because it's what? Flat cells. Okay? Now, these are parts of a cell. You have a cytoplasm which contains the fluid compartment called cytosol, and there are organelles within the cell, within the cytoplasm. Okay? Some of them may have membranes, some do not have membranes. Now, the entire cell is surrounded by what we call a cell membrane, or otherwise known as plasma membrane, right? Okay? And so here is a very nice diagram showing you, or flow chart of the cell made up of the plasma membrane, the cytoplasm. This is the liquid portion called the cytosol, and the one with the organelles has membranes without membranes, non-membranous with membranes. So here you are. It includes the mitochondria, the nucleus, endoplasmic reticulum, Golgi apparatus, lysosome, peroxisomes. Why? Because they're surrounded by a membrane. What about this one? They are not surrounded by a membrane. Your cytoskeleton, microvilli, centrioles, cilia, flagella, and ribosomes. So this is a diagram showing you the structures and the organelles within the cell. So example here, the Golgi apparatus, the mitochondria. If you remember, we talked about this a while ago. It is the powerhouse of the cell because it is where you manufacture or produce what? ATP or energy, right? Yes?
Do sex cells look different than that?
Obviously, yes, they do look different because you already saw the sperm, right? It's like a tadpole, okay? It has a tail of egg, but it has a nucleus, okay? It has all the other organelles here.
Okay.
The only thing is the shape.
Right.
And the other thing about the sperm, if you look closely here, it has, in the cell body here, in the body itself, has all those organelles. It has a mitochondria, and at the same time you have the tail or the flagellum. So the middle piece is actually where you have the mitochondria. So this is a special type of cell called the sex cell. Okay? But in essence, it has all those mitochondria, the Golgi apparatus, all those things. Okay? Now, so here are important things. Before I forget, you are expected to know all these diagrams, and you will learn later on why, because in the lab, the quizzes are based on the Martini diagrams, not the Mariev diagrams. Okay? Example, if I remove this and remove that and put number 1, what's the answer to number 1? Mitochondria. Okay? It's a multiple choice question, but you can answer it right away. Okay? It's like a sausage structure, right? Okay? And you have here rough versus smooth endoplasmic reticulum. The smooth contains ribosomes. What about, the rough contains ribosomes, those dots there. What about the smooth? They do not contain ribosomes. Ribosomes are important for protein synthesis. Again, what are ribosomes for? What are ribosomes for?
Protein synthesis.
Very good. Okay. So again, 4 major functions of the plasma membrane. Again, I do not have to read this. You just can read it for you. 1, 2, 3, 4. Physical barrier, keyword, exchange of materials, exhibit sensitivity to changes, and of course, cell-to-cell communication. So these are the components of the cell membrane: phospholipids, protein, glycolipids, and sterols. Now observe that it is 2 layers. It's called bilayer. Okay? Phospholipid bilayer, because 1 layer, another layer. Observe that there is what? What does fluid here mean? Water here, and what about inside the cell?
Yes.
Water too. What do you call the fluid inside the cell? Intracellular fluid. What about the fluid outside the cell?
Extra.
So what is the opposite of extra?
Intra.
What is the opposite of intra? Which one is fluid inside the cell or water inside the cell? What about the fluid outside the cell? So this is the cell membrane. So the entire cell membrane, everything outside is ECF. What about inside? ICF. Okay? Observe that the phospholipid layer contains the heads facing what? The water. What do you call the water inside the cell? Intracellular. So what is facing the water? The heads. The heads are considered what we call hydrophilic. Hydro means water. What does philic mean? Water loving. Cilia. Now what about the tails? Tail. Hydrophobic. Hydro means water, phobic means they do not like water, which makes sense. If you look closely here, what is facing the water? The heads, because they're hydrophilic, they love water. What about the water inside? What is facing the water inside? The heads. You understand? And why? Because they are hydrophilic too. What about the tails? Hydrophobic. They face each other. They do not want to face what? To water. Very good. Okay. Now there are proteins, channels, intraprotein with channels like this. So you have all these glycoproteins and all the cytoskeleton. The hydrophilic heads are these heads here. So again, I do not have to go over this in detail. Just read this slide here. The hydrophilic versus hydrophobic tail spats towards each other, and then of course you have this glycocalyx. So this is what it's trying to tell us. The heads are water-loving, what the tails are, they hate water. So water is found here outside, extra. Water inside would be what? Intracellular fluid, right? And then you also have all these proteins, but peripheral integral protein. They form channels where substance can go through. Going back to this, see? Go in and out. And of course, you talk about something that's called gated channels. It simply means that they can either open or close the gate so that it prevents some substances to go in or out. See, so again, this is something very important. Get to know this. Some membrane also has with glycolipids and sterols. Now the word permeability. So if the substance can go through or in, out, it's called permeability. Okay? Through the cell membrane. So if it's impermeable, they cannot cross the membrane. So if this is the cell, they can't go inside. Okay. Now what about freely permeable? Of course they can pass through without difficulty. At the same time, selective permeable, that means certain substances they can go through. Others cannot go through because they select which one can go through. Now there are, so study this concept, passive versus active, which one requires energy? Active transport. because it's moving from high to low or low to high? Low to high concentration. What about if it's passive? High to low concentration, what does it mean? If going down here, you do not need ATP, or in other words, you do not need energy. Okay? So these are all examples of passive process, high to low, high to low. But this one refers to movement of what? Water and water alone. What does it mean? The water we're referring to as water. That's the one that is moving through the membrane. And that is called osmosis. So what is the diffusion of water, therefore? Osmosis. Did you get it? What is osmosis a form of diffusion? Yes, it is. But what is being transported? Water and water alone if it's osmosis. Now this one requires a transporter, again, from high to low. So all of these will be what? From high concentration to what? To low. Now what about this one? Active process. From low to high. Does not require ATP or energy. Yes, it does. Now pino and pago are examples of endocytosis, phagocytosis. Pino means to drink water. What about pago? To eat. But what do they normally eat? Especially the white blood cells. Bacteria or foreign organisms. Okay? So here's an example. Now observe that this is what? What does this represent? Carbon dioxide, right? When the concentration of carbon dioxide inside the cell is greater than outside, because you have a lot of these CO2 molecules here, what is diffusion? From high to low. From high to low. Is that an example of passive transport?
Yes.
Yes. Is that called diffusion?
Yes.
And does it pass through a cell membrane or plasma membrane?
Yes.
Yes, it does. Does this require ATP?
No.
No, it does not. Okay? Now look at this. This is this water. This is the solute. Now in a solution, you have two things: the solute and the solvent. In most cases, what is the solvent? Water. The solute could be any, like sugar or what? Salt. Table salt, sodium chloride. Okay. Now, if the solute concentration outside is greater than inside, water molecules will move across the plasma membrane. So, example here, you have a lot of what? Water inside. What will happen? High to low. From inside to outside. Is that osmosis?
Yes.
What is osmosis again?
Diffusion of water.
Diffusion of water. And does this require energy?
Yes.
No, because it's an example of what? Passive transport or processes. Okay? So, facilitate diffusion. Solutes are passively transported across a plasma membrane by a carrier protein. So, example given here is your glucose. This is the carrier protein here with the receptor side. So, the carrier protein is this structure here. In the cell membrane, glucose can go in. Look at the shape there, it matches, and then when it attaches there, then it could go into the inside of the cell. Again, the same thing, high to low, but the only difference is that there is the presence of what? A carrier protein in the wall itself. Okay? So nutrients that are insoluble in lipids or too large to fit through the membrane channels may be transported across the plasma by the carrier proteins, but in only one direction, either into or out of the cell. But there has to be a receptor site there. Now look at the shape there. See, it matches the shape of the molecule. Okay. So again, these are all examples given glucose and certain amino acids. Now, what about active? This is exemplified by the sodium-potassium pump. Very important concept. Okay. So how many sodium are brought out? 3 out. How many are brought in for potassium? 2. 3 sodium, 2 potassium. Does this require ATP? Yes. Why? You're moving from low to high. Okay. High, low. So you see 1, and then there are 3 there. Now what about same thing with potassium? From low, see the yellow things there? 1, 2 compared to 1, 2, 3, 4, 5, 6. So from low to high, you're climbing hill. Low to high. Does that require ATP? Yes, it does. So this is an example of a sodium-potassium pump. How many sodiums are brought out? 3. How many potassiums are brought inside the cell? 2. So this is something. I'm going to share you what happens. When there is not enough oxygen to the cell, You know that you need oxygen for ATP production. Yes, you do in the mitochondria. If there's not enough oxygen or blood flow to a certain organ, okay, so that means what happens to the sodium? The pump is not working, and if the sodium-potassium exchange pump is not working, will sodium be able to go out? No. Where will the sodium go? It remains inside the cell. Why? Because the pump is not working. Why is the pump not working? There is no ATP. Why is there no ATP? Because there is no oxygenation, like when you have ischemia and decreased blood flow to a heart muscle or cell. Okay? So if sodium does not go out, the sodium remains where? And what did I say? Wherever sodium goes, water will
Follow.
Wherever sodium goes, water will
Follow.
So if the sodium does not get out, it remains where?
Inside.
Inside the cell. What happens to the water?
Inside. It goes
The water goes inside the cell, and there be swelling of the cell. Yes, it's called hydropic swelling. From the word hydro, what does hydro mean again?
Water.
Water. So can this cell become swollen? Is that part of pathology? Yes, it will. Okay? It is. Okay. So again, this is an example of endocytosis, either pino with cell drinking. Water and molecules may enter the cytoplasm with mediated transfer diffusion, or phago. What is phago? What is being eaten up? Anything that is considered foreign, like bacteria, viruses, okay? And then it's extruded. This is just, you don't have to memorize the details here. It's an example of receptor-mediated endocytosis. You form this vesicle here. So there's an example of exocytosis too, which is again another form of active transport. So you have all these endo and exo, but you're ejected from the cell. Cellular reticulum vesicles are ejected from the cell. Now, which one is passive? Diffusion, we talked about this already. High to low concentration, it's what we call a concentration. In other words, there has to be a concentration gradient. Something is high, something is low, there's a difference. Okay? And again, what is the diffusion of water? There you go, from high to low, because again, it's an example of diffusion, from high to low concentration. And what about the need for a carrier protein? Still diffusion, but this time it requires what? A carrier protein, okay? And again, glucose and amino acids are examples of these. Again, active transport, sodium-potassium pump, very important. For this pump to work, you need ATP, and in order for ATP to be produced, you need oxygen for ATP production in the mitochondria. Okay? Cell drinking, cell eating. These are usually the cell eating would be the white blood cells. Okay? So pathogens. What are pathogens? They are disease-causing organisms, such as bacteria, virus, and foreign matter. So again, cell drinking, phagocytosis, cell eating. Eating what? The bacteria. That's how you can protect yourself. You talked about endocytosis, receptor-mediated endocytosis. You do not have to know the details here. You don't even have to memorize it. And again, exo is another form of active process. You release intracellular material to extracellular. And again, the plasma membrane can also have all these other projections, like villi. What do you call those villi? These are finger-like projections. What do? They increase the surface area. Okay. So again, these are examples of microvilli there. Now, the cytoplasm, we talked about the need to know the difference between cytosol. What is cytosol? The fluid component of the cytoplasm. These are found there. Nucleus, proteins, and waste. And of course, the organelles, we just talked about them. So the mitochondria, for example, the Golgi apparatus, and of course, you have here the rough or smooth endoplasmic reticulum. Rough contains ribosomes, and the smooth does not contain ribosomes. So again, because of the time limitation, you can just read on the important role played with the cycle, which is, of course, higher concentration of potassium more than outside, okay? And higher concentration of protein, and of course, lower concentration of sodium, because most of the sodium is found outside the cell or extracellular fluid. So either membranous, non-membranous organelles. So take a look at this. There's a table here that summarizes them. Non-membranous cytoskeleton made up of this filament. Some of these filaments you will encounter again in the next chapters, like thick filament of the muscle, thin filaments. So they're actin filament or made of protein again. Make sure you read this slide and understand. Now, I don't expect you to memorize everything here. And there are intermediate filaments like the neurofilaments of the axons and the thick filaments of muscles. Now, microtubules in the cytoskeleton, again, main component of cytoskeleton, can make the shape, change its shape. Central cilia, now the cilia is oftentimes found in the airways. What do the cilia do? They move the mucus. If I were a cilia on the inside of an airway, it will move the mucus, move fluid or secretions across the surface. Now, which one of the cells that we have has a tail or flagellum? Sperm, okay? Now, what happens if you have ribosomes? Again, what is ribosomes for? Protein synthesis. If it's rough, does it contain ribosomes? Yes. See, so ribosomal subunit. So what is the role of mitochondria again? ATP, adenosine triphosphate production. Now, what is the control center of the cell? Where the chromosomes are, control center. Again, these are all the other organelles: endoplasmic reticulum, Golgi apparatus, peroxisome, catalase. So this is an example of the mitochondria here. And of course, we talked about the membranous organelles. They have also the same things as all these other structures that you expect to find in a typical cell. DNA, chromosomes, all these. Now, every time I look at this slide, it reminds me of the crater of the moon. Have I been there in my dream? Okay? But you can clearly see here, it looks like the moon surface. How many of you have been to the moon? Am I the only one in my mind? Okay? You can see here that it's like the craters of the moon, right? All these surfaces. But this is actually what? The nuclear membrane. Okay? The nuclear pores are there. Our DNA, right? So make sure you're familiar with the deoxyribonucleic acid. So half of us comes from the mother, half of us comes from the father. So can you imagine if I was born with blue eyes and blonde hair? What do you think my father would say? I am not your father. It's Darth Vader. Okay? In other words, when I came out, I had turned out with black hair and brown, beautiful, exotic skin. That's my son. Okay? And I got to be like him with a 5'9" height. That's my son. If I was 5'5", that's not my son. Okay? I'm just joking. So apparently, I'm like my father. Okay? I got the height of my father. I don't know where I got my brain. It might be mother or father. It could be both. Okay? Now again, DNA. So you and I have the same. We are Homo sapiens, but you are what you are because of your parents, right? So you should always thank them. Tonight, go home and tell your parents, Dad, thank you for the sperm. Mom, thank you for the egg. If not for your two contributions, I wouldn't be here. On that day, like me, October 20, 1960, I was born 10/20/60 at exactly 10:20 a.m. Does that make any sense? Born 10/20/60, born 10:20 a.m. And in fact, 3 years from now, I will be 60. That will be 10/20/20/20. I'm not kidding. Like, I don't know what happened. 10, October, 20, 20th date, 2020. How many 20s will there be?
3.
So on that day, at 10:20 a.m. on October 20, 2020, I will go to the lottery and I have no idea what happens. But again, I am what I am because of my parents. I did not know it when I look at my birth certificate. 10/20/1960. At 10:20 a.m. Okay? Who delivered me? My aunt, who used to practice medicine in Philadelphia. And in 1960, she was still there in the Philippines, so I cannot forget that fateful day. I was so busy swimming that day, doing my backstrokes in the amniotic fluid, and all of a sudden I could see the scalpel. You know what's a scalpel? Slicing into the abdominal cavity. Oh no! I did not use the word, oh shit. I said, OMG for, I didn't know I would be a Dr. G. I said, I wanted to hide, but I could see the hand of my aunt holding my leg, trying to pull me up. All I could do was, oh hey, oh hey, oh hey. What the heck? What are you doing to me, Aunt? Aunt Chofila? So I could see her signature on the birth of Chofila Sarabia Gamo, MD. And my name became Joel, or Joel, you know. That's my first name, Joel, okay? I just came from Spain, so. Hola! Well, everybody says hola. Okay. Hola la. Now, whenever you say RAF, does it contain ribosomes?
Yes.
Yo, okay? Are ribosomes important for protein synthesis? Yes. So, smooth, do they contain ribosomes?
No.
No. Okay? And therefore, they're not important for protein, but rather what? Lipid, which means fat synthesis. What else? Steroids, carbohydrate synthesis. They store calcium. And what is the meaning of the word detoxification? Getting rid of what? Toxins. Are they harmful or are they harmful? Yes, they are. Okay? So, rough. See those ribosomes there? And smooth. If I ask you for a quiz next week, which part is important for lipid, cholesterol synthesis, and steroid synthesis, the answer would be smooth. On the other hand, which one contains ribosomes? Rough. Which one is important for protein synthesis? Rough. Oh my God! So easy. I love anatomy therefore, okay? It's called positive thinking. So, Golgi apparatus, I think of this as the FedEx. You know, what do? They package. What's the key word?
Package.
Packaging, okay? Secretions, enzymes, all these, right? See, with the vesicles present, okay? So you don't have to memorize the details here. Suffice to say, every time you think of packaging, you think of 1, 2, 3, 4, see? Golgi apparatus, okay? So lysosomes, important to remove bacteria. What is important? They are made up of enzymes that will break down. Okay? So either primary or secondary enzymes. And the same thing with catalase. Anything that ends with ACE is most likely what? In the liver and reperoxysomes. Okay? Now the membrane flow, again, you don't have to know the details here. Transport vesicles or secretory vesicles are present. Now this is a part of a slide that you can read to help you understand in the future, but you do not have to go into the details here. Many cells come permanent or temporary attachment to other cells. So one cell is attached to another cell because of some of these are CAMS, cell adhesion molecules, and again phosphoglycan called hyaluronan. So these are just examples of cell junctions, gap, tight, all these. But again, suffice to say, so one cell can be connected to another cell by these tight junctions. But again, you do not have to know the details here, okay? So what about cell life cycle? Remember, can the cell divide? Yes, your brain cells, your muscle cell, brain cells do not divide. Spinal cord, they do not divide, but bone cells, yes, they do. Your skin, for example, your muscles, for example. The process is called mitosis. So one part of the entire process of cell reproduction. So you have all these prophase, PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. So in the interphase, this is inside the cell, is duplicating G001 S phase. So make sure you understand what each of these phases represent. Duplication of organelles and replication of the chromosomes occurs here. So this is now the parts of after interphase. You have here the actual G1, and this is G0 to G1. So these are parts of the interphase. Replication will occur. Certain enzymes are involved. If you remember the double helix, right? 1 and 2 strands. Observe that what will always bind with adenine would be what? Thymine, see? What is that? Thymine. What is the red one? So A versus T. What about this and this? Guanine. What is this? Cytosine. Always the case. What happens when you have cancer and when there is mutation? This is not being followed. Adenine with thymine. See, this with this. See, this with this. Normally, okay. So again, you either have the different phases from prophase, metaphase. Meta means middle, and then you have all these other cells, stem cells, and telophase, and all these things. See? So observe prophase, metaphase, middle metaphase. They divide and separate. So these are just, again, go over this when you go home tonight, on the next few days, talk about this. Enters prophase, metaphase, anaphase. Now what is significant here is the metaphase, middle of the spindle here. See? And then they begin to separate in the anaphase, and the moment you form a cleavage is called telophase. And the division of two identical daughter cells. If you have 46 chromosomes in the beginning, you end up with 46 here, 46 there. And that is in mitosis. In a different called meiosis is different, okay? In meiosis, sperm is 23, the egg has what? 23 too. So if the sperm fertilizes the egg, 23 plus 23, you get what? 46 chromosomes for human cells. Print off these. Thank you. Okay? Just